ever pondered over the potency of the herbal medicines you consume? How do we ensure their effectiveness and safety? Welcome to the intriguing realm of herbal drug analysis, a vital process that safeguards the potency and safety of the herbal medicines we consume. This is not just a matter of curiosity, but of necessity, as the world increasingly turns to nature for remedies. Herbal drug analysis, however, is not as straightforward as it might seem. The complexity lies in the vast array of compounds that these herbal medicines contain, each with its unique properties and effects. This complexity necessitates meticulous methods of analysis to ensure that the herbal drugs we consume are not only effective but also safe. But how do we navigate this intricate landscape of compounds? The answer lies in sampling techniques, a fundamental aspect of herbal drug analysis. Understanding this requires delving into the fascinating world of sampling techniques in herbal drug analysis. The foundation of any accurate analysis lies in the principle of sampling. But what does it entail? Well, let's dive right in. When we talk about the principle of sampling, we're referring to the process of selecting a small, manageable amount from a larger population. The key here is that the sample must accurately represent the entire group. Imagine you're at a party and you're asked to describe the overall mood. Would you base your conclusion on observing just one person? Of course not. You'd mingle, observe a variety of people, and then form a general idea. That's the essence of the principle of sampling, except we're doing it with herbal drugs. Now let's talk about some critical factors in sampling. First up is sample size. It's like Goldilocks porridge. It has to be just right. Too small and it might not represent the whole. Too large and it might be unwieldy to manage. Striking the right balance is crucial. Next, we have the selection criteria. This involves deciding what characteristics the sample should have to accurately represent the population. In the case of herbal drugs, it could be the type of plant, its age, or even the time of harvest. Lastly, we have randomness. Randomness ensures that each member of the population has an equal chance of being selected. This avoids bias and increases the likelihood that the sample is representative. It's like a lottery, where each ticket, or in our case, each plant, has an equal chance of being picked. So why is understanding the principle of sampling so important? Well, it's the cornerstone of accurate and reliable analysis. It helps us avoid bias and ensures the precision of our findings. In the context of herbal drug analysis, it allows us to make meaningful and valid conclusions about the properties and effects of the drugs based on a small, manageable sample. A comprehensive understanding of this principle aids in avoiding bias and ensuring the precision of the analysis. So let's keep this in mind as we delve deeper into the world of herbal drug analysis. With the principle of sampling clear, the next question is, how do we practically apply this in herbal drug analysis? When we talk about the procedure for sampling, we are looking at a multi-step process. It's like a well-choreographed dance, each move crucial to the overall performance. The steps include selection, preparation, extraction, and preservation of samples. Let's start with selection. This is the first and one of the most important steps. It is vital to choose a sample that is representative of the herbal drug being analyzed. This requires knowledge of the drug, its key components, and where these components are likely to be most concentrated. It's like picking the best apple from a tree. You wouldn't just grab the first one you see. You'd look for the most ripe, the one that best represents what an apple should be. After selection comes preparation. In this stage, the sample is cleaned and prepared for extraction. This can include washing to remove impurities, drying to eliminate moisture, and grinding or cutting to increase the surface area. It's like cooking. You wouldn't toss a whole unpeeled potato into a pot of soup. You'd clean it, peel it, and chop it into pieces. Next, we move on to extraction. This is the process of pulling out the key components from the sample. This can be done using a variety of methods, including maceration, percolation, and distillation. It's like brewing tea. The hot water extracts the flavors from the tea leaves, resulting in a delicious cup of tea. Finally, we come to preservation. This is where the extracted components are preserved for future analysis. This can involve storing the samples in a cool, dry place or using a preservative to prevent degradation. It's like storing leftovers in the fridge. You want to keep them in good condition for when you're ready to eat them again. Throughout each step, there are precautions to be taken to maintain the integrity of the sample. For instance, during extraction, It's essential to ensure that no unwanted components are being extracted along with the desired ones. Think of it as filtering out the seeds when you're juicing an orange. Similarly, during preservation, it's crucial to prevent contamination from external sources. This procedure is not a one-size-fits-all. It needs to be tailored to each herbal drug, taking into account its unique characteristics and the specific components being analyzed. It's like baking. You wouldn't use the same recipe for chocolate chip cookies as you would for a loaf of bread. Each step, when executed meticulously, contributes to obtaining a sample that accurately represents the herbal drug. It's a dance, a symphony of steps that come together to create a masterpiece. A sample ready for analysis. Each step is a note, a beat, a movement, and when they all come together, they create a harmonious melody that is the procedure of sampling. Each step, when executed meticulously, contributes to obtaining a sample that accurately represents the herbal drug. Sampling is more than just a procedure, it carries profound implications for herbal drug analysis. Let's delve into these implications. When it comes to herbal drug analysis, the importance of proper sampling cannot be overstated. It's like a detective gathering evidence for a case. The quality of that evidence can make or break the investigation. For us, it's about ensuring the safety, efficacy, and quality of the herbal drugs. Imagine a scenario where a batch of herbal drugs is sampled incorrectly or inconsistently. The implications are significant. If the sample taken doesn't accurately represent the entire batch, the analysis could be skewed. This could lead to incorrect conclusions about the safety and efficacy of the drug. A drug perceived as safe might actually have harmful side effects, or a drug thought to be effective might not work at all. The implications extend to the trustworthiness of the results. When we talk about herbal drugs, we're dealing with people's health and well-being. It's paramount that the results of the analysis are reliable and trustworthy. Proper sampling lends credibility to the analysis. When done right, it ensures that the results are representative and accurate. It builds confidence in the drug's safety and efficacy, encouraging more people to consider herbal remedies as a viable treatment option. Sampling also has broader implications on the herbal drug industry. For manufacturers, proper sampling can help ensure product consistency, which is crucial for brand reputation and consumer trust. For regulatory bodies, it provides a reliable method to monitor and control the quality of herbal drugs in the market. In essence, sampling is a powerful tool in the world of herbal drug analysis. It's not just about taking a small part of something larger. It's about understanding that small part so we can make accurate conclusions about the whole. It's about ensuring that the herbal drugs we use are safe, effective, and of high quality. The power of sampling lies in its ability to shed light on the hidden complexities of herbal drugs. So, what have we unraveled today about the world of herbal drug analysis and sampling? Well, we've delved deep into the intricate realm of herbal medicines, which have been used since ancient times and continue to be a vital part of healthcare worldwide. We've learned that the efficacy and safety of these herbal drugs hinge on thorough analysis and precise sampling. Let's recap, shall we? We kicked off by exploring the principle of sampling. Remember, the goal of sampling is to collect a representative sample that mirrors the characteristics of the whole lot. This is crucial as it allows us to make informed decisions about the entire batch based on the analysis of just a small part. We discussed stratified random sampling, systematic sampling, and cluster sampling, each with its unique approach and application. Next, we dug into the procedure of sampling. We learned that it's not as simple as just picking a handful of the product. It's a meticulous process involving several steps, from pre-sampling activities like defining the lot and determining the sample size to sampling activities like collecting, labeling, and preserving the samples, and finally post-sampling activities like transporting and storing the samples. Each step is vital to ensure the integrity of the sample and thus the accuracy of the analysis. And then we looked at the implications of these sampling techniques in herbal drug analysis. We saw how they ensure that the herbal drugs we consume are safe and effective. By detecting contaminants and verifying the presence of the claimed medicinal components, sampling plays a pivotal role in quality control and assurance. So there you have it, a whirlwind tour of the complex yet fascinating world of herbal drug analysis and sampling. The next time you consume a herbal drug, you'll appreciate the science that ensures its safety and efficacy. Subscribe for more because trust me, you won't find me later.